A study by the CDC found hospital-acquired infections cost the United States between $10 and $17 billion in 2010. Even a marginal reduction of these infections, with associated mortality rates between 12 and 25%, will be cost-effective in addition to saving lives. A high frequency of infection is associated with the use of invasive devices, in particular central lines, urinary catheters, and ventilators. So how are surfaces kept clean from bacteria in a hospital environment? The answer is through the application of solutions of formaldehyde, phenols, or cationic surfactants. Solutions of these compounds are applied to medical devices and other surfaces which often require re-sterilization. However, bacteria have advanced defense mechanisms to combat disinfectants. As communities of bacteria collect on surfaces, they form a protective matrix called a biofilm. These biofilms often cause infection and become increasingly challenging to combat with commercial antimicrobial disinfectants. Antimicrobial agents can damage the outer layers of the biofilm, while high concentrations of disinfectant may never contact the inner cells. This leads to antimicrobial resistance. Bacteria which survive the disinfectants will continue to grow and divide stronger than before. When the community of bacteria reaches a certain population, they trigger virulence, and disease is the result. Recent developments of antifouling coatings, or slippery coatings, aim to reduce the initial attachment of bacteria. My research in the Schiffman group focuses on new techniques to prevent the initial attachment of bacteria so biofilms can be prevented. Specifically, I work on adding antimicrobial functionality to current antifouling coatings in order to improve the long-term effectiveness of slippery antifouling surfaces. Through the use of patterning techniques, strips of antifouling and antimicrobial coatings will be placed next to one another in order to study their effects on bacteria attachment. Implementing the universal coating technique on medical devices will replace the need for currently used cationic surfactants, reducing bacterial resistance in a hospital setting. Why fight bacteria after robust biofilms have been formed? My name is Eric Rice, and I am a senior chemical engineering student at the University of Massachusetts Amherst.